go ahead and get started and thank my co-investigators for the trial up front. It's a multicenter trial sponsored by Celgene. Oh, hello, sorry, I'll start over. My name is James Kochenderfer from the NCI. I'm presenting a multicenter trial of anti-BCMA CAR T cell product known as BB2121. The trial is sponsored by Celgene, and up front I'd like to thank my co-investigators in this multicenter trial who are all listed here. First of all, BCMA, a promising target for multiple myeloma. Clearly, multiple myeloma is a disease that is essentially incurable, and new treatments are needed desperately. BCMA is a member of the TNF superfamily. It's expressed universally on multiple myeloma cells, and critically, expression is restricted to plasma cells and only a very small subset of B cells. Most B cells do not express BCMA. We've shown in a phase 1 study at the NCI initial proof of principle for the anti-BCMA CAR T cell. This was published in Blood in 2016, Ali et al., and there's an abstract today at this meeting. Brudno et al., it's actually abstract 524, will be presented this afternoon. T cells transduced on these studies were transduced with a gamma retroviral vector encoding an anti-BCMA CAR with a CD28 costimulatory domain, and significant cytokine release occurred on these studies. The trial that I'm talking about now is with our multicenter trial. The T cell product called BB2121 utilizes a different CAR. This CAR is encoded by a lentiviral vector and contains the same single-chain FV, which is right here called 11D5-3. It contains a 4-1BB costimulatory domain and a CD3 zeta T cell activation domain. We showed in preclinical testing this CAR was highly effective in vitro with low tonic signaling and was able to eradicate solid tumors of multiple myeloma in mice. Our protocol design started out with low doses and escalated up in a 3+3 manner. We started with a dose of 50 million total flat dose CAR T cells, CAR positive T cells. Then we went to 150 million, 450 million, then 800 million. The general protocol schema was for leukapheresis, and then manufacturing, which took 10 days plus the release testing. Chemotherapy was given as a conditioning regimen to enhance the activity of the CAR T cells. Fludarabine 30 milligrams per meter squared, cyclophosphamide 30 milligrams per meter squared. CAR T cells were infused on day 0. Follow-up was conducted with blood draws and bone marrow biopsies. First response assessment was done at week 4. The data which I'll present in detail today is about our dose escalation part of the trial. These are the data that we have prepared and certified for presentation. 24 patients. Clinical deterioration occurred in 3, which led to us only dosing 21 of the patients. And we have 21 of those patients, all 21 evaluable for response. Manufacturing success rate was 100%. Since these patients were treated and follow-up was completed to a certain degree, we've treated 12 more patients on an expansion cohort. I will not present these 12 in detail today because the data has not been fully analyzed yet. The baseline demographics of our first 21 patients, data cutoff October 2nd, 2017. Median follow-up 35 weeks for these patients. Age 15 to 8. ECOG performance status was 0 or 1 for all patients because that was a protocol requirement. A large fraction of the patients had high-risk cytogenetics, 43%. One very critical thing about this study is how heavily pretreated these patients were. It was an incredibly heavily pretreated group. 7 was the median prior lines of therapy. 100% of the patients had a prior autologous stem cell transplant. 100% had bortezomib, and 67% were refractory to bortezomib. 100% had lenalidomide, and 86% were refractory. 91% had pomalidomide, and 71% were refractory to that. We have here the various combinations, common cumulative exposures. 100% were exposed to bortezomib plus lenalidomide, and 67% refractory. You can see the other combinations, 91% were exposed to bortezomib, lenalidomide, and carfilzomib, with 48% refractory, so a very heavily pretreated group. Our adverse events, generally this was a very well-tolerated CAR T-cell product, especially in comparison to other protocols that I participated in. 71% had cytokine release syndrome, but only 10% was grade 3 or above. We had some neurological toxicity, 24%, but 0% of that was grade 3 or above in the dose expansion cohort. The neurotoxicity was generally much milder and less prevalent than what I've seen in previous anti-CD19 CAR studies. We also had neutropenia, thrombocytopenia, and anemia. No dose-limiting toxicities were observed in the dose escalation. Cytopenias were mostly related to the flu-cy conditioning. We did have some slow recovery of counts, which is somewhat to be expected in multiple myeloma patients with very low blood counts and extensive myeloma involvement in the bone marrow. We had 5 deaths. 3 of these were due to disease progression on the lowest dose level of 5 million CAR T cells. We had 2 patients who had what we call the active doses, which was greater than 50 million CAR T cells that died. 1 died of MDS and 1 died of cardiac arrest. 14 patients experienced 1 or more SAEs. Many of these SAEs were due to our strict protocol SAE requirement, where a patient who was admitted only with grade 1 or 2 CRS or fever was counted as an SAE. This slide summarizes our responses. You can see, I mentioned before, the patients on the lowest dose level of 50 million did not have good outcomes with progressive disease in all 3 patients, but after that, in the active doses of 150 million, 450 million, or 800 million CAR T cells, we had 17 out of 18, 94% of patients had an overall response. 10 of the 18 patients obtained a complete remission. So just to look at this, the green is complete remission, 10 complete remissions. Another important point is at the 52-week mark, we had 5 of these patients so far that have met the 52-week, 1-year progression-free survival standard. We have durable ongoing responses over a year now, ongoing in many patients. Our longest response that's ongoing is 68 weeks at this point. Responses continue to improve as late as month 15, where we had a very good PR to CR transition. Median progression-free survival has not been reached in the active dose cohorts, and again, we call the active dose cohorts 150 million and above. So in general, very impressive responses compared to my previous experience treating multiple myeloma. I want to emphasize that in our patients now, we have somewhat more mature data over the past 6 months, which shows a deepening of responses over time. First of all, time to first response was 1 month median. Time to best response was 3.74 months. Time to complete response, 3.84 months. Duration of response and progression-free survival not reached. Importantly, the durability is shown here with a progression-free survival rate at 6 months of 81% and a progression-free survival rate at 9 months of 71%. These 2 graphs show the deepening of response over time. We can see here at as of May 4th, 2017, with n equals 15, we had a 27% CR rate. And if we look at October 2nd, 2017, where we locked our data for this presentation, we had a 56% CR rate. So the responses deepened. We did lose 1 responder to progression during that time, so we went from 100% response rate to 94%. But the responses going on deepened with the shift from PRs and very good PRs to CRs. Again, very indicative of durability of these responses. So in summary, we've shown durable responses with BB2121 at active doses of 150 to 800 million CAR T cells. we had durable response in a very heavily pretreated population. 94% overall response, 89% very good PR or better, 56% CR or better. Progression-free survival not reached. MRD negative results were seen in 90% of the MRD evaluable patients. Disease progression in 4 patients occurred. 3 of 3 evaluable patients remain BCMA positive at progression. To date, safety profile has been manageable, with doses as high as 800 million CAR T cells. 2 reported events of grade 3 CRS resolved within 24 hours. So I've talked mainly about our dose escalation because we blocked our data on that, but I do need to mention 1 patient in our dose expansion phase who had a delayed onset reversible grade 4 neurological toxicity. So again, this is on the dose expansion phase, and we're not fully reporting the dose expansion phase, but in the interest of full disclosure, we are going to disclose this 1 grade 4 event which occurred in our dose expansion phase, grade 4 neurologic toxicity associated with tumor lysis syndrome and CRS. Patient recovered completely, the neurologic toxicity resolved. The patient had the highest disease burden on the trial, and the patient has now obtained a very good PR despite low BCMA expression on their myeloma cells. Finally, we have a trial that we're starting up, a global pivotal trial called KARMA, which is open to enrollment, and the BB21 dose range will be 150 to 300 million CAR T cells. Thank you.